So I should, first of all, start by saying thank you very much for everybody who's listening to this presentation, whether you're listening to it now as I'm giving it live on November the 14th, or whether you listen to the recording. So it is being recorded from the beginning, and we will make it available on our website, hopefully by tomorrow morning, technology permitting, but as soon as we can. But yes, thank you very much for your time and for your interest in the Canine Genetics Centre. I'm expecting this talk, my slides, to last about 45 minutes. And then after that, we will take a break. We will turn the camera off and have a comfort break, and also go to give us a chance to go through some of the questions that you can submit from now. There's a QR code at the bottom of this slide, and it appears on a few of my other slides throughout the talk. So if you would like to send us a question, we will do our best to respond to it at the end. But I don't anticipate we'll have time to get through all the questions. So we will take our time about answering them, and those would also be available on our website as soon as possible. But we will prioritise putting the recording up. But the answers to those questions will follow as quickly as we can, hopefully within the next week or 2 weeks, that sort of time frame. Okay, so I think we'll, with that, having said that, we'll crack on with the main body of the talk. So my talk is going to go something along these lines. I'm going to start with a research update. I'm going to tell you about our peer-reviewed publications over the last 12 months. And then a little bit about our current areas of research, or our main projects that we're working on. Then I'm going to use a couple of slides to talk about other highlights, other news, other things that we've been getting up to over the last 12 months. I will then give a fundraising update on what's been going on over the last 12 months, and then what our plans are for 2025 and beyond. And there's that QR code again, if you would like to send us a question. So peer-reviewed publications. These are really important for researchers because they show that our findings are robust. They position them in the public domain so that other researchers can take advantage of them or use them. And they demonstrate to funders that we undertake publishable, robust research. So for those of you who are not familiar with the peer review process, I'm just going to whiz through it really, really quickly. So when we get to the end of a project or complete the project, or at a good sort of halfway point, we will write up our findings and we submit those to an appropriate scientific journal for peer review. The journal will then send the manuscript out to at least 2 experts for anonymous review. Those experts return their reviews to the journal, including, required revisions and recommendations that they think the authors should make. The authors then get the opportunity to amend the manuscript if required, resubmit it. Sometimes there might be several rounds of those revisions that are requested. But finally, hopefully, that manuscript is accepted for publication. It could, of course, be rejected. And then those results are placed in the public domain at that point. That whole process takes usually about 6 months. And that's on top of the time it takes to actually do the research in the first place. So when you get to the point of publication, that's sort of an important step in the research process. So in the last 12 months, we have published 5 papers in peer-reviewed journals. So this is just the first one that was published in, I think, November or December of 2023. And just to sort of talk you through, so a citation like this, the authors will come first. So I've highlighted in bold here the authors from the Canine Genetics Centre. The other people who are listed are other collaborators, other researchers from other institutions, or some ophthalmologists on this paper. So this was the paper that we published describing the mutation that causes multi-ocular defect in the Old English sheepdog. And at the bottom, you can see here a URL, a web link, and that takes you to that paper on the journal. So the journal is PLOS ONE, and this is how that paper appears on that journal. So up here on the right-hand corner, you can see that 1,300 people have viewed this paper. It's been cited once, and if you wanted to, you could download the PDF here because, as I say, we try to publish exclusively in open access journals. The second paper to be published this year was one describing a mutation in the English shepherd that causes progressive retinal atrophy. This story had very good publicity. It was featured in the New York Times, actually. And that's because it was the story started with an English shepherd called Shola, who was a search and rescue dog. And she went blind, so she had to be retired. And I think it was a very sort of emotive story because it illustrates very well that, behind all these research projects, there are real dogs who are suffering from these inherited diseases, but also the people involved in the lives of those dogs as well. So I think that's why this story hit the press to the extent that it did. This paper was just myself and Jonas Donner, who works for Wisdom Panel. And what we did here was we analysed the frequency of two variants, one in a gene called RPGRIP1 and one in a gene called MAP9. These cause or are associated or modify progressive retinal atrophy in lots of different breeds. We've been working on these variants for probably about 20 years. RPGRIP1 variant was found in Dachshunds originally, but now we know that both these variants occur in many, many breeds. And so this was a multi-breed analysis and that we were able to do because we teamed up with Wisdom Panel, who have vast data sets. And I'll be saying a little bit more about that later on during my talk. This was a paper that I published with two ophthalmologists, just looking at the prevalence of an eye disease called ocular melanosis in the Cairn Terrier. And last, but by no means least, a paper that we published studying a paroxysmal dyskinesia in the Norwich Terrier. And this was a preliminary genome-wide association study. And our findings were published in Animal Genetics just about 6 weeks ago. We have 3 additional papers that are currently under review. So these have gone through some of that peer review process, but have not been accepted for publication yet. The first, idiopathic epilepsy in the Italian Spinoni. We hope that, so that's already been through review. We've addressed the revisions and it's been resubmitted, and hopefully that will be published literally any day now. The second one, describing hereditary cataract in the Siberian Husky. Again, that's gone mostly through the peer review process. And then the third one is a paper describing the mutation that causes a retinopathy with vitamin E deficiency in the English Cocker Spaniel. So this is quite an exciting story. So we're calling it RVED, used to be called central PRA or CPRA. And this actually is a treatable inherited disease. So owners of homozygous dogs, so dogs that have 2 copies of this mutation, should talk to their vet about vitamin E supplementation, because we think in most dogs, restoring vitamin E to normal levels should prevent symptoms developing in most cases. And so this is an example of a DNA test that will benefit dogs directly rather than preventing the disease in future generations, which is how most DNA tests have their benefit. So this is quite an exciting story for us. And initial findings from the research suggest that possibly up to 200 Cocker Spaniels might be affected with this disease in the UK each year. It's a disease, I should say, that along with the retinopathy, there are sometimes neurological signs that go with it. And so this disease is sometimes mistaken for other conditions. So now I'm going to just go through our current areas of active research. Inherited eye diseases, these have always been a mainstay of the Genetics Centre, and we are currently investigating, multiple different diseases in multiple breeds. Idiopathic epilepsy is a focus of ours as well. We are currently investigating this in depth in 7 different breeds. Intervertebral disc disease in Dachshunds and in Cocker Spaniels. We are at any one time always sort of investigating several, what we think are probably monogenic or genetically simple diseases. At the moment, that includes PHPT in Keeshans, a novel neurological disorder in Labrador retrievers, and congenital vestibular defect in Welsh Springer Spaniels. I'm going to talk a little bit about the Welshie project on my next couple of slides, but I don't have time to go into the other two in great detail. Other complex diseases, or probably complex diseases that we're investigating, include steroid-responsive meningitis in beagles and histiocytic sarcoma in flat-coated retrievers. And again, if you'd like any more details about where we are with those projects, do contact us afterwards. So I'm now just going to spend a couple of slides talking about our inherited eye disease research, and I'm going to talk about all the programs of work in fairly broad terms, just to try and keep this presentation to a manageable length. So our inherited eye disease project is known as CRIDE, and this stands for Consortium to Research Inherited Eye Diseases in Dogs. And that's our logo. And this was a project that was initially funded by Dogs Trust back in 2019. That block of funding has now come to an end, but we are still, as you will see, we've still got a lot of data that was generated with that funding that we are working our way through. So this is now, widely recognized, and we, get DNA samples from dogs that are affected with inherited eye diseases, from veterinary ophthalmologists and from owners and breeders, and they come to us from all over the world. And what we did at the beginning of the project, and I'm going to explain this on the next couple of slides, is to design a workflow that we use to screen these affected dogs. And these are dogs that are affected with any eye disorder that is likely to be inherited. So we screen DNA from each dog for all the published inherited eye disease variants. So those are all the mutations that have been through that peer review process that I talked about at the beginning of the talk, and that are in the public domain. And we do this in large batches to make things efficient and cost effective. When a new variant is published by ourselves or by other researchers, then we update that screening panel, and we also update the CRIDE database. So this is the front page of the CRIDE database. This is a publicly available, open access, searchable database, and includes details of over 20 different diseases. I think just over 100 published mutations now in lots of different breeds. And so you can use the search box, to put in the breed that you're interested in or the disease that you're interested, and it pulls up all the details of all the variants that have been found that match those search criteria. So it's a very useful resource, we think, for veterinary ophthalmologists and for dog breeders as well. So this variant panel, I say, is currently just about 100 published variants, and we screen the DNA from every affected dog for all of those variants. And doing that gives us information in a couple of main areas. First of all, it will tell us if a known variant is segregating in a breed of dog that it hasn't previously been reported in. And it also tells us if it's likely that a novel or an unknown variant underpins the disease that we're investigating at that time. So this is just a little sort of very simplified version. So I've written here 5 different variants, but the list is actually, as I say, about 100 long. And every DNA sample, whether it's, a Labrador Retriever with PRA, or a Nova Scotia duck-tolling Retriever with lens luxation, or a Tibetan Terrier with PRA, or a Cocker Spaniel with multiocular defect, they will be screened for every variant. Most of the dogs that we screen do not have those variants. Occasionally, we will get a positive. So, for example, this is a hypothetical example, but a Labrador with PRA having the PRCD variant. So that would not be a surprise. But for example, if we screen a Tibetan Terrier and it doesn't have this variant at the bottom in FAM161A, then that might tell us that this dog has a novel variant. And it then progresses into sort of the research pile and gets investigated further. So the initial stages of this project were spent on designing and testing that inherited eye disease variant panel. It's now up and running and works very well, and we have used it to screen several hundred dogs. And it's served to, identify dogs that don't have what we call a molecular diagnosis. So they don't have any of known variants. We've gone on to sequence the whole genomes of just under 80 dogs, so 79 dogs of 33 different breeds that are affected with what we suspect could be as many as 19 genetically different diseases. We've then, so analysing those data, that's led us to the variants, 3 of which I've already mentioned, multiocular defect in Old English sheepdogs, PRA in English Shepherd, retinopathy in Cocker Spaniels, and another one in the Italian Hound that's a novel eye disease that's also associated with sort of morphological abnormalities as well. As I've described, we publish all our findings and then go on and develop DNA tests that then can be used to eliminate or eradicate those diseases from those breeds. So there's a huge data set left to analyse. As I said, we've said 80 dogs nearly we've sequenced the whole genomes of. We're analysing those data as quickly as we can, and we would, funding permitting, aim to sort of update our bioinformatics tools to help us accelerate that process if we can. So that's a broad overview of our inherited eye disease work. Essentially, we're sort of investigating many different diseases in many different breeds in parallel. So now I'm going to move on to talk about idiopathic epilepsy research in the CGC. So this is work that is led by Sally Ricketts in the group, and the aim of which is to identify sets of genetic variants that are reproducibly associated with epilepsy in a particular breed. So epilepsy is a complex disease, so we don't talk about variants that cause epilepsy, but we use this word association for complex diseases. And what we would really like to do is to develop genetic risk scores, polygenic risk scores, that can identify variants in a population that are at the greatest genetic risk of disease. So if you look at this little graph over here, if this is genetic risk along the bottom and this is population, many complex diseases have a normal distribution. We would like to be able to identify the ones that are at the right-hand side and that are at the highest genetic risk. And then that information could be integrated into breeding tools. By identifying genetic variants and understanding the genes that they are located in would give us a better understanding of the biological processes that underlie epilepsy, and that could hopefully lead to better treatments and better diagnoses as well as preventative tools. So to date, we have focused on border collies, and there's a paper in preparation describing our findings to date. The Italian Spinoni, as I said, that paper is hopefully very, very close to publication. A little bit of work on the Keeshond and a collaboration with the University of Helsinki on epilepsy in the Labrador retrievers. So we have a new genetic study that started at the beginning of this year, that's taking a whole genome sequencing approach to identify those genetic risk variants in 5 new breeds. And those are written here: the Beagle, the English Springer Spaniel, the Giant Schnauzer, the Hungarian Vizsla, and the Irish Setter. And this is funded by the Pet Plan Charitable Trust. It's a 2-year grant that started at the beginning of 2024 this year and will goes to the end of 2025. And this grant is covering project consumables and whole genome sequencing and some genotyping, and is covering the full-time salary of Chris Jenkins, who is a postdoctoral member of the team, part funding for Sally, who is the principal investigator, and Jim, who is the idiopathic epilepsy study coordinator. And those stakeholder breeds have contributed significantly to the funding of this project, and we are very grateful for that. Between them, they have contributed over £22,000, and that's been used to fund sample collection and part of the Jim's salary as well. So this is just a little timeline. So ongoing, we are currently still collecting DNA from cases and unaffected dogs. So the cases have to be collected very robustly to make sure that they are genuine idiopathic epilepsy cases. And so we work very closely with veterinary neurologists. We have sequenced the whole genomes of 5 cases and 5 unaffected dogs of those 5 breeds that are listed at the top. That data is due back shortly. Once that has arrived back in the lab, then they will be using, our computational pipeline to analyze those data and to identify candidate risk variants. And those candidate risk variants are then validated by genotyping in much larger cohorts, ideally 100 cases and 100 unaffected dogs from each breed. So that's why sample collection is always ongoing pretty much, and is a big part of what we do. And then in addition to those 5 breeds at the top, we will also be undertaking sort of smaller scale analyses for additional breeds for which we have perhaps 1 or 2 whole genome sequences from cases. They might be breeds that contributed to our Give a Dog a Genome project that dated back to when we were at the Animal Health Trust. So the final sort of research program that I'm just going to mention is intervertebral disc disease. So this is work that we have begun really since we came to Cambridge, and we collaborate very closely with the neurologists in the hospital here on this work. So intervertebral disc disease is a very common cause of spinal disease in dogs. Clinical signs, they vary. At best, they're painful, and at worst, they lead to complete paralysis of the limbs, which can be irreversible. So intervertebral disc disease, it's a considerable welfare concern. So work of others has identified a genetic risk factor for IVDD, which is an overexpressing FGF4 retrogene on chromosome 12, better known to most people as CDDY, and DNA tests for CDDY are widely available now. So many of the shorter-legged dogs or chondrodystrophic breeds have got 2 copies of this variant, and therefore those breeds are more likely to develop IVDD than other longer-legged breeds. But despite the fact that many breeds share this genetic risk factor, there are differences between breeds in how they present with IVDD, the typical age of presentation, and also between individual dogs. There are differences. And by no means all the dogs that are homozygous or have 2 copies for this variant develop IVDD. So this sort of suggests that there are additional genetic risk factors involved. And a lot of our work is centered around trying to find those now. So the IVDD work is undertaken by Bruno. So Bruno Lopez, he's a veterinary neurologist from Southfields, but he's studying for a part-time PhD based at Cambridge Vet School. So when he's here, he's embedded in the Canine Genetics Centre, which is really useful, actually, and it's very handy to have a vet and a neurologist sort of in our midst, because most of us are obviously geneticists. So we're taking a whole genome sequencing approach, currently sequencing the genomes of Dachshunds with IVDD and Dachshunds without IVDD. As I said, to try and identify some of these extra variants that are associated with the disease in this breed. Analysis, the genomes have been sequenced, the data is ready, and we will start to analyze that shortly. And that work has been funded by CamVet, which is the Cambridge Vet School's own charity. We are currently collecting DNA from Dachshunds that have been assessed under the Kennel Club Dachshund Health UK IVDD screening programme. So they have a calcification score. And in partnership with Wisdom Panel, we will shortly be investigating those dogs to try and identify regions of the genome that are associated with that disc calcification score as well. So that's work that will start shortly. And then finally, we have a pilot project, and this was funded by the Kennel Club Activities Health and Welfare subgroup. And this is to look at the disease in Cocker Spaniels, because Cocker Spaniels are at risk of IVDD as well. And we're looking to see if there's a relationship between body size and genotype of this CDDY risk factor. So that's a bit of a whirlwind tour of our main programs of research. And I think what I'd really like to stress is that the Genetics Centre exists for all breeds. A very frequently asked question is, what is the Canine Genetics Centre doing to benefit my breed? And that's a very understandable question. So the research that we're undertaking, benefits, has the potential to benefit the health of multiple different breeds. But I think it's also very important to understand that the Genetics Centre is here to help investigate new and emerging diseases which can occur in any breed at any time. And to illustrate that, I'd just like to show you, talk very quickly about this paper. This was not our work. This was done recently by a consortium that are sequencing the genomes of 10,000 dogs, actually. And what they did was look at the de novo mutation rate. So that's new mutations in 43 different dog breeds. It's new. It was published in June of this year. And what they did was they sequenced trios of dogs, so both parents and one offspring, 404 different trios. So this is a very large scale sequencing project. And their findings were really interesting. So one of their findings was that, the effect of paternal age per year on mutation rates is about 1.5 times greater in dogs than it is in humans. And that one of the mechanisms by which mutations can occur, recombination, is 10 times larger in dogs than it is in humans. So essentially, the message is DNA mutates. This is normal, occurs in all breeds. We can't stop it happening. By no means all the mutations that occur are deleterious, but some will be, and some will cause or be associated with an inherited disease. And because breed dogs represent isolated populations, those disease variants can become widespread and much more can spread through a population much more quickly than they might do in, a more outbred human population, for example. So I've just got one slide here where I'm going to give you an example of one of these potentially emerging diseases that we're currently working on. So the disease, we think, is a congenital vestibular defect in Welsh Springer Spaniel puppies. This came to our attention within the last year, and this disease has not been reported in this breed prior to this litter, to our knowledge. So 2 affected puppies out of a litter of 9, born to seemingly healthy parents. The pups were very obvious from birth. They were very wobbly. They continuously just sort of rolled over and over, and they vocalized a lot. They were very, what you might describe as squealy. It was quite distressing to listen to them, actually. We were sent videos. These dogs have improved, apparently, with time, although they may have some residual health issues. So they may not be completely normal, as it were. So the breeder was very engaged. We collected DNA from the affected puppies, from the unaffected littermates, and from the parents. The work of that sequencing, the sequencing that we did was ring-fenced from ring-fenced donations from the breed community. And so we've sequenced the genomes of that whole family, and the analysis is currently underway to identify that causal variant. So we hope we can find that variant, develop a DNA test, and that disease can sort of be eradicated before it ever really becomes a problem. If you'd like to read more, just we featured this as one of our news articles on our website, so you can find it there if you're interested in reading a little bit more about that. So that's our research, really, or the sort of a whirlwind tour of research. And now I'd just like to spend a few slides talking about some of the other activities that we have been up to over the course of this year. So very big news for me personally was that in July, I was very, very honoured to be awarded the Kennel Club International Canine Health Lifetime Achievement Award. I was the co-recipient of one of the other awards, the International Award in 2015. So I was really very humbled to receive the Lifetime Award as well. I was nominated for the award by my peers, and that made it, all the more special, really. But I had to acknowledge I was there with my award, but the success behind that was really due to many very loyal and hardworking people in the Genetics Centre team who have made that the work sort of possible, all of which have got a passion to improve the health of dogs. So I really consider this a group award, really, rather than a personal award. But it came with a very, very welcome $50,000 cash prize. And so that has also made an invaluable contribution to the research that we've been able to do this year. So some other things that we were up to, we went to the International Conference on Canine and Feline Genetics and Genomics in Helsinki in June. So this is the foremost conference really for dog and cat geneticists. It happens every other year, and so it was really great to be able to go. Chris Jenkins from the group was invited to give a talk. So there's Chris giving his talk summarising some of the epilepsy work. This led to some sort of excellent conversations with collaborators over the course of the 3 or 4 days. And again, this was paid for by a ring-fence donation to the Canine Genetics Centre. So we were very very grateful for that. In other news, in March, we went to Crufts, and that's a photograph of me getting a very big check from the Old English Sheepdog Health Group. So that was very welcome. In July, on a very blustery day, Sally and Jim went to the Beagle Welfare 45th Anniversary Fun Day, again, presented with a cheque. Not quite as big as the Old English Sheepdog one there, but just as gratefully received. In August, we attended the Kennel Club International Agility Festival to measure Cocker Spaniels. That was for the pilot project that I mentioned for IVDD research. In September, 4 of us did the London Big Relay to raise funds for the Genetics Centre and for epilepsy. Between us, we ran a half a marathon, and that was a tiring but a good fun day. And just to show that we are not all about work, one of our team members, Ellen Schofield, represented the UK at the Flyball World Cup. All of these stories and many, many more are featured on our website. We have a really well-populated news section. We try to put a post out every single week up on the website. We also post it on our social channels. And once a month, we issue a newsletter in which about 4 of these news items are sort of pieced together and emailed round. And these articles are a variety of things. So we have the Welshie story, for example, the Norwich Terrier paper. This article down at the bottom was how we handle our enormous data sets. We post funding updates and sometimes just information articles. So this was one that we published about breeding with carriers. So I would really encourage you to head over to the website. There's a lot of work being put into our website over the course of the year, and I think it's very, very informative now. So now I'm going to turn my attention to fundraising and give you an update on what our financial situation is at the moment. So in January of this year, I gave a similar presentation to this one. And during that presentation, I reached out to our grassroots stakeholders, really, asking for support to keep the Canine Genetics Centre running. And I appealed to our supporters to help raise, a pretty substantial £345,000 to keep the Genetics Centre team together for the following 12 months, to basically buy us some time to secure some longer term and more secure funding. To ally concerns regarding security of funds, quite understandably, breed clubs and individuals were given the opportunity to pledge funds in the first instance. And then the Genetics Centre agreed that we wouldn't request those funds until we had collectively raised enough funds to keep the Genetics Centre going initially for the first 3 months until the end of June 2024. So this is a summary of our fundraising. So pledges from breed clubs who took advantage of that pledging option, we raised about £80,000 through that. I think that was raised by about 90 different groups representing about 42 different breeds. And then we also received direct donations from individuals and clubs to the tune of about £127,000. So that total came to £207,000, which never in my wildest dreams did I think, when I gave that presentation in January, that we would raise anything like that amount of money. We've had other income. So the Lifetime Achievement Award, that was just under 40,000. A couple of weeks ago, I got a donation from the British Association of Veterinary Ophthalmologists for 6,000. We've had a little bit of grant funding. So our total income from this year from donations and other sources is £284,000. A few adjustments. We did unfortunately lose one member of the team, so that reduced our payroll cost by just over £50,000. But we have had to meet some other non-salary expenses of about £42,000. So essentially the funding gap to cover the running costs until the end of March next year has been reduced from £345,000 to £52,000 currently. And those funds have been raised in a myriad of different ways. So donations have been made by individuals from all over the world, from UK, from Europe, from the USA. Donations and pledges have come from literally hundreds of different clubs and societies and organisations. And these are your traditional breed clubs, to breed rescues, to agility clubs, training clubs, ring craft groups. If you can think of it, I'm sure one of them has made us at least one donation. There have been some really creative ways of fundraising. Some clubs have added amount to show entries and donated those to the Genetics Centre. People have recycled ink cartridges, held quiz nights, held seminars. Some of the ways have been very energetic. So Joanna Bowman ran 2 marathons in a week, believe it or not, and raised just over £2,000. The Health and Rescue Foundation of the PBGV Club of America, they have they made a very, very generous donation of $10,000 to the centre, but they have also offered up an additional $10,000 as a matched fundraising. So for every dollar that's donated, they will donate another dollar. So that's a very good opportunity for anybody considering a donation to effectively double their funds, and we're just about halfway there. And then just at the beginning of this week, I heard about this festive fundraiser from Amber Marie that's currently raised nearly £200. So these are all, some of the, amazing and creative ways that people have been working so hard behind the scenes on behalf of the Genetics Centre this year. So I just want to take a slide now for thank yous. People normally do their thank yous at the end of talks, but I don't want to forget them, and I don't want people to sort of wander off and make a cup of tea and not hear all the thank yous that I have to give. All our donors, each and every one of you, wherever you are and however much you donated, thank you, thank you, thank you. Not only donations, but we have been literally overwhelmed with messages of support this year. So again, thank you to everybody who has got in touch and told us how much the work of the Genetics Centre means to them, because that means a lot to the team and has sort of fortified our resolve, I think, to find a way to secure the future of the Genetics Centre. I need to say a very big thank you to Ben Stanbury, who designed our new logo that's down at the bottom left of all of these slides. He designed that logo free of charge for us. So I definitely recommend, if anybody needs any new logos or branding, to head over to Ben's website. He was so helpful and a great talent. I've had lots and lots of conversations and lots of advice from people all year, but these 7 people listed here have spent literally hours and hours talking to me, talking to each other, working behind the scenes, filling in spreadsheets with contact details, coming up with ideas, helping me to understand how the purebred dog world works, because I'm not in that world. So thank you very, very much to all of you. I would like to thank Our Dogs newspaper, who have given us a lot of exposure right from the beginning of the year. We're incredibly grateful to you. Thank you. The University of Cambridge Vet School and the philanthropy team who have got behind us and supported us. All these donations have caused an awful lot of work for people working on the gift registry and in the accounts team. I'm very, very happy to give them all that extra work, but it has meant extra hours for all of those. I'd like to thank the genetics team, my own group of people who have worked so hard this year in times of, uncertainty, to not only do all the research that I've talked about in the first half of this talk, but also to do all the extra work that comes along with basically running a big fundraising campaign. So thank you very much to all of those. Our funders, Pet Plan Charitable Trust, CAMAVET, the Departments of Veterinary Medicine and the Activities Health and Welfare subgroup for funding. Thank you very much. Thank you very much to customers of CAGT. That is our own commercial genetic testing service at the University of Cambridge, all the profits of which are redirected to the Canine Genetics Centre and help support our research. They are now making a consistent surplus each month. So thank you very, very much to everybody who has given them your custom as well. Just a big thank you, really. I feel that word is not enough to express how I feel, but everybody who has put efforts in to keeping the Genetics Centre going. Just thank you from the bottom of all our hearts. So we are still here, thanks to all those efforts. So I now would like to talk to you a little bit about what happens from the end of this year and beyond. So in January of this year, I did promise that I would not ask breed clubs for donations on the same scale again, and that the large sums of money that I was requesting were really emergency funds to keep the Genetics Centre running while we tried to secure longer term and more secure funding that obviously was a result of us losing our long-term funding that we had had for quite a while. So this has been a major focus of 2024. Certainly for me, I have had many conversations and emails, etc., to try and secure funding. Funding opportunities have remained limited from animal and veterinary charities, which have been sort of the mainstay of much of our funding in previous years. That's just a reflection, I think, on the fact that many people are still struggling with cost of living and have maybe less money to donate to these charities. I've had discussions with numerous potential funders, have started many, and some are still ongoing, and I hope to be able to provide more details about those shortly once they have been come to fruition and been finalised. But I can share details of one agreement that has been confirmed, and I can do that right now. So the Canine Genetics Centre is very excited to announce that we have agreed a 2-year research agreement with Wisdom Panel. So we've collaborated informally with Wisdom Panel for many years now, but we have now signed a formal agreement. So Wisdom Panel will provide support for 3 members of our staff over the next 2 years. And they will share with us genetic data from their really extraordinarily large data sets of genotyping data from breed dogs and from non-breed dogs and crossbred dogs. And we will have access to some of those data, and we're very excited about that. And then Wisdom Panel are also going to support some of our IVDD research that I went through a few slides ago. And in return, we will provide our sort of genetic expertise to help explore those data sets, to gain new insights and to direct canine genetic testing in the future, as well as some consultancy and advocacy for genetic testing and how it can help improve the health and well-being of dogs. So I am very excited about this agreement. I think it's a huge opportunity for us to have access to data on a scale that we would not be able to generate ourselves. And this is a picture of Oliver, who is the Genetics Research Director from Wisdom Panel, but many of you will have known Oliver when he worked with us at the Animal Health Trust. So we are, I think both very, very excited to have this sort of formalised agreement now and be able to sort of work together much more closely going forward. So stakeholder donations. So research is expensive. It costs about £400,000 a year to run the CGC. And I say about because it's a bit, how long is a piece of string? The more money we have, the more research we can do, and the more different approaches we can try. And I think, although, we have an agreement in place with Wisdom Panel, and we have some other potential agreements in place, and we will continue to apply for every potential grant funding that we can. I think it's obvious to me that donations from grassroots stakeholders will remain very important for the foreseeable future. And so we would really like to, at this stage, encourage regular giving from individuals, clubs, societies, and various dog groups. Regular giving provides us with regular and predictable income, and that enables us to plan ahead. And whereas I'm not sitting here and telling you that I don't want large one-off donations, we absolutely would be grateful for those as well. It's this idea of regular giving that enables us to sort of, plan ahead with a bit of predictability. And of course, we can claim gift aid on donations from UK taxpayers, which increases those donations by 25% at no additional cost to the donor. And another frequently asked question that we have is, will my donation be safe? And that's where I think that one of the benefits of regular giving is that by donating a little and often, it does provide the donor with some security. So a direct debit can obviously be stopped at any time if the circumstances of the CGC changes or if the donor's circumstances change. So I would like now to introduce you to a new regular giving scheme that we are calling a Friend of the CGC. So I would really like to invite you and your friends to consider becoming a friend of the CGC, which is easy. We just ask you to set up a monthly direct debit for £5 a month or more if you can afford it, and allow us to claim gift aid if appropriate. And that's it. To thank you, as a small token of our thanks for becoming a friend of the CGC, we will give you a lapel pin. It will look something like this, that we would hope that you would wear with pride, and maybe a starting point for conversations to spread the message about the CGC and the work that we do. And as another token of our gratitude, we will ask you, you'll have the opportunity to nominate somebody for the new Friends of the CGC Health and Welfare Award. And I came up with this idea because, as I said, when I was nominated for my Lifetime Achievement Award this year, the fact that I was nominated by my peers was really significant for me. So we would like to give you the opportunity to nominate somebody for an award so that you can sort of help other people feel appreciated. So this award is going to be sponsored by CAGT, our genetic testing service. And as I say, you'll be given the opportunity to tell us about somebody that you know personally or you know of who makes a positive impact on the health and welfare of dogs. And that could be anybody. It could be your breed health coordinator, but it doesn't have to be. It could be your local vet. It could be somebody who runs a rescue centre. It could be your local dog groomer or your local dog trainer who just does a good job for dogs. And the winner will receive their award at the next research day. We held one of these research days in September of last year. It was very well received, and so we'd like to hold one in the spring of next year in Cambridge, between Crufts and Easter, the date to be confirmed. And we very much hope that the winner, or the winners, if we get lots of nominations, we may be able to give more than one award. We hope that they will be able to join us, but if they can't travel, then we will send them their award, and we will record them, and we will play that recording at the research day with their permission, obviously. But we hope that that's something that, you know, might be something that grows in scale and in future years. I'd like to think it might. So I'm getting to the end of my talk now. I see I'm cracking on to 8 o'clock. So this is just my last slide. So I'd really like, I think, to emphasize the idea behind this regular giving is that, you know, lots of small donations add up, and people don't have to give a lot. But obviously, the more people that give, the more money we would receive. And I think what we've seen over the course of this year is that there are a lot of supporters out there for the CGC. When I gave my presentation in January, about 600 people listened to it, either live or listened to the recording. So I hope that this presentation will be listened to by that sort of number of people. And if we could get 500 friends signed up as a result of this presentation, that would be amazing. But if all of those people could persuade at least one other person to become a friend, that would be 1,000 friends of the CGC. And if half of those friends could donate £5 a month, which is, you know, not far off the price of a cup of coffee or a price of a bag of fancy dog treats, and maybe half of them could donate £10 a month, that would generate us an income of £90,000 a year. And if we could claim gift aid on 1 in 4 of those donations, that would actually generate us just shy of £120,000 a year. And that, added to our agreement with Wisdom Panel and other discussions we're having and other grant funding, that would actually be, you know, a game changer for us to have that level of regular income coming in that we could depend upon. Would make, I think, all the difference to our financial stability over the coming years. So I've talked for a little bit longer than I intended to. I think now we will have a 5-minute comfort break. We will come back and we will try and answer as many questions as we can. We will answer the questions that have been asked by the most people, if we have any of those. In the meantime, if you'd like to head over to our website, please do. Here's our website. The button up here that says support us, that will actually take you to become a friend of the CGC direct debit page. So if you'd like to have a read of that, then please do that now. Here's the QR code to submit a question, and I'll leave it there for now, and we'll be back at bang on 8 o'clock, hopefully. So see you in a minute or two. Okay, so I think hopefully people can now see me. I don't know if that's a good thing or not, and more importantly, hear me. So I think, yes, because it is 8 o'clock now, so I don't want to keep you from your things you need to do the rest of your evening any more than I have to. So we have had some questions in. I'm going to answer, I think, about 3 or 4 of them now, and then I will, as I say, we will go through them and post the answers to the rest of them on our website as soon as we can. So we've had a couple of questions on asking which breeds we focus on, which diseases we focus on, and how different breeds might work with us to develop a DNA test, for example, for a condition that they are concerned about. There's a variety of factors that go into that. The diseases that we study, the disorders that we study, they all have to be sort of serious and debilitating. There needs to be engagement from the breed, largely for our success. There's no point really us deciding that we want to investigate disease X in breed Y if that breed, or at least a subset of that breed, stakeholders aren't interested in the work that we're doing. So that plays a big role. Having clinicians to collaborate with us, that can be a really important factor as well. And then funding, you know, that same old thing again. You know, we do need funding to fund the research that we do. So all of those things, I would say, if you are from a breed, you have a particular health concern, please talk to us. You know, send me an email or go onto our website and use there's a contact us form that you can fill in and start that conversation. You know, we'd be very happy to understand more about the diseases that are of concern to different breeds. We had a question about publications and the fact that we publish these scientific publications, but you know, they are scientific in nature. So I would say, so certainly papers that are published by the Canine Genetics Centre, those news items that I was talking about, they are essentially lay summaries of those sort of synopsis of the papers that we've published. So whenever we publish a paper, we will publish shortly after and one of those news articles, which will be a sort of slightly less detailed version of the paper. So please, again, go to the website and sign up for to receive notification of those news articles, or follow our Facebook page as well. We always publish when we make a news item available, we publish it on our Facebook page. Lots of questions from Keeshans, owners and breeders out there about PHPT. This is, I didn't talk about it in detail today, but this is a very active area of research for us. We are aware that the DNA test that was offered by Cornell is no longer available. And we are now collaborating very closely with Claire Wade from the University of Sydney to work on this. And so we have, you know, we're really sort of putting some effort into it. We know where the mutation is, it's a very tricky part of the genome to work with. So we've tried some sort of different ways of sequencing that region of DNA. And we, you know, really hope that within the next few weeks to, you know, maybe a month or so, we'll have some positive news for you about that. And certainly, as soon as we are able to offer a DNA test from the University of Cambridge, we will do that at the very earliest opportunity. And the final question that I think I'm going to answer this evening was about epilepsy. People were asking, or somebody was asking about when we find variants, genetic risk factors for epilepsy, are they the same within breeds, or does each breed have its own set of genetic risk factors or variants or mutations? The answer is we don't really know at this stage. As I said, epilepsy is complex, so it's not just one mutation in a breed, apart from potentially very rare examples. So we expect each breed will have multiple genetic risk factors or risk variants. Some of those may well be shared between breeds or occur in genes that are in common pathways between breeds, but it is also likely that each breed will have its own set of sort of private variants as well. But the more we tease apart the genetics of epilepsy in different breeds, and the better our understanding is, the more we will understand what the genetic basis of that disease is across breeds. So I think I'll leave it there as far as questions go. Once again, thank you very, very much to everybody who has listened. If you didn't get the opportunity to listen to all of it, or you know people who didn't manage to listen to it tonight, please head to our website. Hopefully by tomorrow, technology willing, and you'll be able to listen to the recording. And yeah, thank you very, very much for listening, and have a very good evening, everybody.